Good morning, everybody. Already we learned about what is pilot plant and what is the scale up and how it is converted into a large production and what are the objects involved in pilot plant scale up. All those things we learned in last class. So today's topic is development process for the formulation. So what are the steps involved in this development process means different steps it is involved. So developmenting of the batch laboratory scale. So first we have to develop a laboratory scale. Okay, after that pre-exhibit batch or the process qualification batch we have to exhibit. So this is the before going to a exhibit batch, that is nothing but it is a practical batch production. Before going to a production we have to exhibit, and after before the exhibit we have to pre-exhibit the batch and process qualification batch we have to develop. After that the exhibit batch is nothing but the pivot batch. The next step is the exhibit batch. Next one, these two we call it as a scale up batches. The pre-exhibit batch and exhibit batch we both we call it as a scale up batches. Next one, next step is the production scale up batch. So what are the different batches we are conducting in developing and development of the process formulation means first development batch we have to produce in a laboratory batch. Already our research and development we are conducting. So after that pre-exhibit batch or process qualification. So for the qualification of the process, for the running of a process, what are the qualifications we need? For that we have to design one rough idea, and for that we are conducting the pre-exhibit batch. So after that exhibit batch we are performing. After that the production batch. Okay. Coming to the laboratory scale batch, how we are performing this laboratory and what we are performing in this laboratory scale batch. Early, it is a early development of the laboratory batch or a research and development batch. So it is generally 100 to 1000 times less than the production batch. Already you know that we are developing in a small quantity. Okay, so that small quantity is 100 to 1000 times lesser than the production. In production batch, how much the volume? It will be in lakhs, or in it will be in thousands. But in laboratory scale up, it is only it is very less quantity we are preparing. That means 10 to 10,000 times it is lesser. So what is the use of this laboratory scale batch? To support a formulation. First, to support the formulation we have to prepare a R&D scale. First we have to prepare in small quantity. If it is works, then we have to go for the large quantity. So next one is the packaging development. So how we have to package this after formulation? So which package condition is suitable for this drug? That also we will know about by this. Know about this packaging development. Next, preclinical trials also we are conducting. Okay, this in this laboratory scale only the preclinical trials we are conducting. Okay, after that pre-exhibiting batch. So this is the first scale up batch. So it is the first scale up batch. So it is generally 70% of the size of the exhibition batch. So for the full production, so out of 100 it is 70% it is larger than the laboratory scale batch. Okay, and it is less than the 30% of the exhibit batch. Okay, so it is prepared with the complete process documentation and fully validated analytical methodology we are doing. In this batch, what we are doing, complete process documentation and fully validated analytical methods we are conducting. Okay. Next one is the exhibit batch. So this is the last step of the scale up batch. It is a demonstration batch. That means it is a practical batch. Okay, practical demonstration batch. It contains a manufactured documents and production specification, acceleratory stability data submitted to the FDA. So what we are doing in this batch, not only the production, we are documenting how we are manufacturing, that is documenting, and product, what are the product specification, what are the standard specifications, and how the product is specifying, how the quality control and purity control, how it is giving the purity, all the product specification, all the details about the product we have to specify. And not only that, stability, accelerated stability data also submitted to the FDA. After this batch only we are submitting the stability data. Already you know that why we are conducting the stability, to check the check the physically, chemically, the product is stable or not within the environmental conditions. Not only the environment, but the accelerated stability also we have to check. So it is generally 10% of the production scale batch. So it is almost near to the production batch, but it is a 10x is nothing but the 10 times it is bigger than the first batch. Okay, it is manufactured under a GMP. In this process only we have to consider the good manufacturing practice, the guidelines we have to follow, and with the guidelines only, the equipment guidelines and production guidelines and manufacturing guidelines, everything we have to follow according to the GMP practice only. So this exhibit batch, also call it as a, it is having different names. Okay, if they in your exams they are asking differently, means don't confuse on that. So it is having different names. So what are the exhibit batch, also call it as a NDA or ANDA batch. Why we are conducting? After this it is going to the preclinical studies, preclinical studies and the clinical studies, it is going. So everything will be that's why we call it as a NDA batch and ANDA batch. Okay, and not only that, regulatory references batch. It is a this batch exhibits the regulatory reference. After this it will all the specifications will be mentioned in the for the approval of the FDA. All the information were submitted to the FDA with the application of the NDA and ANDA. So it is a NDA and ANDA are the branches, one of the branches of the regulatory affairs. That's why we call it as a regulatory reference batch also. So next, bioequivalence batch, that is nothing but in ANDA, in generic product development, bioequivalence studies we are conducting for branded drugs. What we are conducting, biological animal test and the then the animal test and the bioavailability test, PK, pharmacokinetic test we are performing. But in bioequivalence batch, what we will conducting, we are comparing with the standard or the reference drug or the marketed drug we are comparing. That we are called it as a bioequivalence. We are equally referencing with them, comparing with the marketed product. That's why this batch also called it as a bioequivalence batch and a fully GMP batch. That why it is called it as a fully GMP batch? It is following the GMP guidelines. That's why we also call it as a fully GMP batch. And stability test batch, why we are telling that this is a stability testing batch? Because we are conducting the stability accelerated stability test. Why we are conducting? To predict a shelf life and also the packaging material, which packaging material is suitable for the product AP dosage form, and also the determination of the expiry date. So all the things with this only we are applying to the FDA. So that's why we also call it as a stability tested batch. Coming to the production batch, it is produced during the routine marketing production marketing of the product. So while conducting, this is the running production batch. So it is 100 times larger than the pre-exhibited batch. So this is it is almost 300 to 1000 kgs, or 300 to 10,000 liters, or 1 lakh to 3 lakhs to 3 lakhs to 10 lakhs unit. So it is 100 times bigger than the pre-exhibit batch. Okay. Scientific scale up. While doing a scientific scale up, it is very, very important. So what we have to do in this scientific scale up should done the incremental manner, and the process being validated for each new scale. So when you are developing a new scale, it should be validated. Validated means the process, everything, the process should be documented. For what purpose? Means to submit to the FDA. So why we are submitting the FDA? We want the drug approval. If we want to launch our product to the market, that needs a approval from the FDA. FDA approval is very, very important. So what are the factors considering for approval of this new drug development? Means it should be fully documented version. FDA is reviewed, and this after reviewing of this documentation versus only it will give permission for the approval to market. So the process, whatever the process you are conducting, that should be documented for each scale. Each scale you have to document it. What you are performing, everything should be documented. So the equipment should be processed following similarity. If you are performing for the equipment, the equipment wise, what we have to consider means the geometrical similarity, kinematic similarity, and dynamic similarity. It should be follows. That means geometrical means the shape wise and size wise and length wise, everything it should be similar to the what the GMP guidelines they are referring. That should be suitable, or matches with the GMP guidelines. And not only that, kinetic element means speed, rate also it should be same to the, and dynamic wise also it should be similar. Okay, these are in detail we studied in further next classes. So we will not only that, in MPharmacy or PhD you will know about this in detail process. So in general consideration, very, very important, what are the for pilot plant scale up, to develop a pilot plant scale up, what are the general considerations, what are the considerations we have to generally take? So first one is the reporting responsibility. Very, very important, reporting responsibility. Who will having the reporting responsibility? R&D group. First it is divided into two groups. Okay, two subtypes. So first one is the R&D group with a separate staffing group, and staffing group, and it manufacturing the industry, all the way it should be separated from the formulation section, and it will be from the formulation sector within the plant only, because of the confidential research work. So the research work should be separated from the staffing work, because it is having the confidential work. If we release it, means our new, it will not consider as a new drug till the marketing it reaches to the market. It should be a confidential, then only no one will copy from for your product or processing. Okay, so that for that it should be separated from other staff. So next one is the formulator should develop the product can into the production and can provide support after transition into the production has been completed. So this R&D, the R&D group, it is, even though it is separated, it should be contact with the formulator. So whatever the formula they are giving, that should be implemented. Okay, implemented by the formulator only. So if he is facing any problems or any critical situations, or for smooth running of the production also, it needs the R&D guideline, and also the formulator, the communication should be always present, not only with the R&D to the formulator. It should be communication, the good communication with the staffing, other staffing groups and marketing team also. It should be good communication, and also accounting also it should be good communication. So if with each and every department, that should be reported should have the reporting responsibility, then only the product efficiently pass from the these FDA guidelines, FDA rules and regulations. So this is the reporting responsibility. So in each stage, in each department, whether it is a pass or fail also you have to document. So all those things are considered by the FDA. Next, coming to the personal requirement. Personal requirement is nothing but the scientist with a, so who are the persons are required, are suitable for this pilot plant scale. Personal requirement is nothing but the who are the persons are suitable for this pilot plant operations. That is very important. So the scientist with the experience in pilot operations, as well as the actual production areas, the most preferable. That means the experienced persons are only suitable for the experienced and scientific persons only suitable for the pilot plant operations, not only that, for the production also. So then only if they are facing if any problems arising while conducting operations, already they are experienced, so they will rectify by with their experience. They will solve those problems easily. So as they have to understand the intent of the formulator, as well as the understanding of the perspective of the production personals. So whatever the formulator intended, that should be communicated. Okay, and the production personals, so that should be whatever the production personals needs and the formulator needs. Okay, that should be understand. When he will understand, if if he have the knowledge only, he will understand what they are telling, what they need to improve their quality, okay, improve their product quality. So if they are suggesting formulator or the production personals, if they are suggesting any suggestion, that should be understandable. So when he will understand, when he is experienced, then he will understand. So the personal requirement should be with the experienced persons only, the personal or the experienced. So the group should be have some personal with the engineering knowledge, as well as the scale up also involving the engineering principles. That means who are conducting that person should know the knowledge about engineering of the equipment. If suddenly while conducting sudden while conducting a production, if any critical situations arising with the equipment or running equipment, immediately solved by this scientist. So the not only that, the formulation, for that formulation, which equipment is suitable, and which equipment, which size of the equipment is suitable, and how much the production should be, the space, how much space should be there, everything will be calculated by this scientist only. So he should have some knowledge about in engineering, engineering principles of the equipment, and also the space and everything. He should get some knowledge. Okay, he should have some knowledge. Next one is the space requirement. How much space is required? So what is the space requirement for the space, for what purpose space is required? For the administration and for the administration and the information process and the physical testing area, we need a space for the physical testing, we need a space for the standard equipment, and we need a space for the storage area. So these are the space requirement. It is very, very important. In administration area and information, we will learn. Okay, what is the administration and information process? In this, adequate office and desk space should be provided for the both scientists and technicians. For administration work also, we need some space and desk space. He should report to the higher authorities. How he will report? He should be, it should be documented. So we have to provide some space to do some work. For written work, we have to give the desk space. With the desk space only, he will documented. So for that is reason, we have to, for administration purpose and information purpose, he should, we have to give some adequate space. Not only that, so whenever the, if anyone comes to the for the formulation purpose, if anyone wants to meet this person, or the anyone wants to meet with the scientist, where he will meet, at the while production area he will meet? No. So he will have some desk space. At that space, they will come and they will communicate with the scientist. So the space should be, the space should be adjacent to the working area. It should be not too long for the working area. Otherwise, he will get some difficulties for to conduct a documentation work. It should be nearer to the working area. Okay, physical testing area. So here physical testing should be provided permanent benchtop space for the routine used physical testing equipment. So here, what are the physical testing here? The general means a tablet, weighing machines and pH meters and dissolution apparatus. These should be permanently fixed on the benches. So that space, the top space on the bench top, it should be permanently fixed. So that's why we call it as a physical testing area should be permanently bench top face. Okay. Next one is standard pilot plant equipment floor space. So here the discrete pilot plant scale, where the equipments needed for the manufacturing all type of dosage form is located. So whatever the equipment for all the dosage form, whatever it may be, the different, for the different doses form, the equipments should have some space. For all the doses form, what the equipments we are using, for all the equipments, the some space should be some space should be required. So that should be located in a discrete pilot plant space. In intermediate batch, it is nothing but the full scale production equipment is essential for the evaluation effects of the scale up of the research formulation and process. That means the full scale production, the equipment should be essential for the evaluation. This is the intermediate batch, intermediate pilot plant scale. So the permanent equipments are permanently fixed on this process, means a large volume of the equipments are installed. So for the installing this equipment, we need a large full scale production. We need a intermediate stage space. Next one, equipments used should be made portable wherever the possible. So after use, it can be stored in the small store area, small storeroom. So some equipments should be portable. So large equipments we can't be portable, it should be installed. But some equipments should be, after completion of this production, we have to move that equipments to the storage area. So for that also, it should be, equipment should be portable. So the space for that also, we have to require some storage space, and the space for the cleaning of the equipment should be also provided for the cleaning purpose. So we are not providing any space for cleaning, how the equipment will be cleaned? So it will leads to contamination. That's why we have to give some space for the cleaning of the equipment and the personals. Okay. Next one, storage area. So storage area. In this storage area, these are divided into approved and unapproved areas with the active ingredient as well as the excipients. See here, I want to tell in detail in this storage area. Actually, storage area is very important for the production. So what we will storage. So raw materials we are storing, and the approved products we are storing, and unapproved means rejected products also we are storing, and tested compounds also we are storing, and finished products also we are storing. So all the products we are storing in the storage area. So how it is a storage area, how we are differentiated? So first, whatever the raw material we are getting, that will be that will be stored in a raw material storage area. Okay, after that the QA person or QC person will come and take the samples for testing of the quality and purity and efficiency. For that they will take, so at the time they will give one tag to that raw material. So that raw material is that tag is in yellow color, and the product that product will be shifted to the yellow area. Yellow area is nothing but the quarantine area. Quarantine area is nothing but it is under the testing condition. The yellow area is nothing but it indicates that it is in testing process. So after the testing process, the QC person will come. If it is approved, he will change the tag. Okay, that in the tag, it will contain the product, and that is in, if it is approved, that green tag he will tag to that raw materials. So that green tag contains drug ingredient names and size, and the quality, how much percentage it is containing the purity and quality, and everything that is that will be in green tag, so that after approving this raw material will be shifted to the approved area that will be separated in a green line. Okay, that approved products are separately kept in a green area. That green area is nothing but the approved product. So unapproved product is nothing but these are tagged with a red tags. Okay, in this, with this red tag, means unapproved raw materials. This raw material will be shifted to the rejected area. From this rejected area, it will be again sent it to the return to the original manufacturer for the wholesale manufacturer, where we get this raw material, we are sending to that person. So different areas should be provided the storage. Not only that, of the finished product, after finishing the packaging also, the materials should be shifted to the storage area. So why in production area, the raw material and the approved raw material only should be stored. So how much the quantity needed, that raw material only should be present in the production area. Okay, so remaining product, after finishing this production, immediately it should be shifted to the storage area. So where this finished product is shifting, means the finished area, finished storage area we are shifting. So the process, in process material and finished bulk product from the experiment scale up batches made in the production, same thing. So storage area for the packing material should be provided. So packing materials also, we have to give some storage area. Okay, not only the finished product, we have to give the packed materials also, we have to give some storage area. Okay, coming to the review of formula. So a thorough review of each aspect of the formulation is important. So in the formula should be the formula should be thorough review for the formulation purpose. So the purpose of each ingredient, so you should know what is the purpose of each ingredient. Okay, then only you will know about what it contributes to the final product manufacturing, what that product is plays an important role in manufacturing of the dosage form. That we will know. On the small scale laboratory equipment should be understand. In small scale only we will know about the review of the formula. So in R&D only you will know about the review of the formula importance, each ingredient importance, and what it contributes and what it leads to the product manufacturing. That you should know the thorough knowledge. Then the effect of the scale up using the equipment that may subject the product to the stress of the different types and degrees can be more readily be predicted or recognized. That means the scale up, the effect of the scale up of using the equipment may be subjected to the product. So for the to stress the different types and degrees of more readily predicted, recognized, and if any stresses, any difficulties we are facing, that can be rectified. Review of the material. So materials, how we will review these materials. One purpose and responsibility of the pilot plant is approval of validation of the active ingredient and excipients raw materials. So the materials, whatever you are using, the materials that should be approval and that should be validated. Not only the active ingredient, excipients also should be validated. Okay, not only active ingredient only we should not validate it, excipients also we have to evaluated and the documented. It should be in documented. So whether why we have to do, if excipient having any toxic effects or contaminations, it will ultimately affect to the active ingredient. So the ultimate dosage form will be affected. So entire production it will be affected. So the raw material used in the small scale production cannot be necessarily be the both representative for the large scale production. So uh, so raw material used in the small scale cannot be, whatever you are using in small scale, that everything should not be related to the large scale production. So we have to consider some raw materials for the small scale production also. Next one, equipments. So the most of economical and simplest and efficient equipments which capable of producing product within the proposed specifications are used. So what if for the efficient equipment, for the efficient output quality of the dosage form, for what we need efficient equipment we need, that should be in a simple form and that should be in economical wise we can afford. Okay, so the cheap low cost, it should be in low cost and it should be simple and it should be efficiently it should be work. That should be for the better producing a product within a proposed specifications. So the size of equipment should be such that the experimental trials can be relevant to the production size batch. So those equipments should be the for the experimental trials also, the equipment should be in a suitable position to handle that equipment for the trial batches also. Okay, that equipment should be like that. So suppose if you want to prepare 10 then means 1000 tablets means 1000 tablets means that that equipment should be suitable for 1000 tablets and that equipment should be suitable for the 1 lakh production also. So that the equipment should be in a trial experiments and also the production size batches also. And if the equipment is too small, the process developed with the not scale up. If too small means the scale up will not be proceeded. That's why it should be in a medium sized. Okay, it should be for the small scale purpose and it should be the large scale purpose we have to use. So it should be too small, it should not be too large. Okay, it should be in a intermediate size. So whereas if equipment is too big, then the wastage of the expensive active ingredients, if what will happen if it is too large means the wastage will be happen. Okay, the already the active ingredient is very expensive. If the wastage leads to loss of production. Okay, so it will leads to it will leads to loss of profits. So quality wise also it will be decreased. So production rules. What are the different production rules we have to follow? The intermediate as well the future market trends and requirements are consideration determining the production rate. The production rate, it is intermediate as well the full market trend requirements. This production how it should be it should be future trend markets and present trend market it should be reached. Okay, whatever we are doing, the production rate determine while determining the production rate. So process evaluation, very very important. This process evaluation means the after while conducting a process, what are the parameters? Evaluation is nothing but to check that product or the compound or the drug product within the specified parameters or not, we have to check. So what are those parameters here? What are the parameters here? The mixing speed, how much speed, and the rate addition granulating agents of parameters, and mixing time, and heating and cooling rates, and filter size, and screening size, and drying temperature, and the drying time. Very very important. If drying time is too small, means it will give some defects, and if it is too large, means it will give some defects. So drying and mixing, if we improper mixing, also improper mixing speed, if the mixing speed is increased, also it will lead to some production defects. It will leads to production defects. So the in order of the mixing of the compounds also we have to check. So all these things we have to check for the parameters, whether it is within the limits or not. So next, ninth one is the master formula process procedures. So what we have to conduct in this weigh sheet and processing directions, and also manufacturing of the procedures, we will conduct in manufacturing, master manufacturing procedures. So this one we will study in, we will in detail I will explain in next class.